Hello, my name is Neke Ibeh, and I'm excited to tell you a little bit about myself. I currently work as a senior bioinformatician at the Princess Margaret Cancer Center, and my work involves applying computational and statistical methods to questions in cancer biology. But before I get into describing what led me to this particular career path, I'd like to shed some light on my background. I'm Nigerian, and my siblings and I were born in Bulgaria since my parents were studying there. I immigrated to Canada with my mom and my siblings when I was 8 years old. For as long as I can remember, there was always a major emphasis on academics in our household, and I took a very strong liking to the medical sciences early on. My interest in disease dynamics was really sparked during my undergrad as I completed a degree in mathematical biology. Mathematical biology is a rapidly growing research area, and some of the most popular subject fields include the modeling of behavior in animals and plants, for example, life history trade-offs, parasite-host dynamics, and speciation, as well as population genetics and evolutionary ecology. Mathematical biology allowed me to use a multidisciplinary approach linking theoretical and experimental biology. As an undergraduate student, I had the privilege of learning how to explore the significant role that mathematics and statistics play in modeling the evolutionary dynamics of infectious diseases. Personally, I was very intrigued by the development of models and their use either as predictive tools or as a means of understanding fundamental epidemiological processes. So, I pursued a master's degree in bioinformatics in order to be able to further delve into such themes. Bioinformaticians use computational tools to analyze biological data. The field of bioinformatics allows us to combine knowledge from biology, computer science, and statistics. As a graduate student, I was able to continue exploring this intricate relationship between the evolutionary dynamics of diseases and statistics using various computational algorithms and tools. During this time, my main project explored the Ebola virus and its evolution, specifically how the 2014 variant reached unprecedented transmission and mortality rates. This Ebola project yielded some really interesting data as it showed that the complex interplay between weak selection, epistasis, and structural constraints shaped the evolution of the 2014 Ebola virus variant. This hadn't been explored before, and it shed some light on the role of selection in the emergence of the 2014 outbreak. Here, selection refers to a process whereby the host's immune system would determine which strains of the virus are able to survive and replicate. Epistasis within and among genes is defined as the effect of one mutation being dependent on or correlated to the presence or absence of one or more different mutations. We then decided to further expand these findings, and we explored the evolutionary networks that existed in the influenza and Zika viruses. Now, as a bioinformatician working in the field of cancer research, although I'm no longer studying evolution as it pertains to species or viruses, there's still a lot of parallels. Cancer development within an individual is also an evolutionary process, which in many ways mirrors species evolution. Species evolve by mutation and selection, acting on individuals in a population, and tumors evolve by mutation and selection, acting on cells in a tissue. On our team, we handle genomic data from a very wide range of cancers, and the biological questions being answered really depend on the system that's being studied. I have personally been focusing on a branch of analysis whereby we're able to analyze the cancer samples on a cell-by-cell basis, as opposed to studying the bulk tumor tissue sample. This approach is called single-cell analysis, and it has really facilitated the study of cancer heterogeneity, and it has also allowed us to better understand the mode of action and the proliferation of various cancers. Using such approaches in a computational framework, I have been able to explore evolutionary processes and themes such as tumor suppression, drug and therapy resistance, as well as cell diversity and proliferation. As I work towards obtaining my PhD, I hope to continue exploring the mechanisms that drive the evolution and progression of human disease.